I'm going to talk on the topic phosphorylasis of glycogen and starch. Before discussing the topic phosphorylasis of glycogen and starch, let's describe what is phosphorylasis. Phosphorylasis is a type of chemical reaction in which a phosphate group is used to break a bond in a molecule. Okay, this is a chemical reaction in which an inorganic phosphate is added to a molecule, and that phosphate can act as reagent to cleave a bond in that molecule. This reaction is catalyzed by a different type of enzymes called phosphorylase enzyme. And phosphorylasis is totally different from hydrolysis because in hydrolysis we use water to break a bond, while in phosphorylasis phosphate group is used to cleave a bond in a molecule. Means in hydrolysis water acts as reagent, while in phosphorylasis phosphate acts as reagent. That is the difference between phosphorylasis and hydrolysis. Going further, a lot of us are getting confused between phosphorylasis and phosphorylation, but these two terminologies are totally different from each other. Phosphorylasis, as we discussed earlier, is the cleavage of bond by using phosphate. Okay, this is a breaking reaction, while phosphorylation is an addition reaction in which phosphate is attached to a molecule. Usually ATP is added to a molecule. Okay, this is the difference between phosphorylasis and phosphorylation. Now we will discuss the topic phosphorylasis of glycogen. Phosphorylasis of glycogen is the enzyme catalyzed breakdown of glycogen by inserting an inorganic phosphate molecule at glycosidic bonds, releasing glucose 1-phosphate and shortening the glycogen chain. Now let's describe more. We know that in glycogen, okay, glycogen is what? It is a polymer of many glucose molecules, okay, interconnected through glycosidic bonds or glycosidic linkages. When an inorganic phosphate is added to glycogen, then these glycosidic linkages break down, okay, and one end of glycogen is replaced by that inorganic phosphate, and it produces glucose 1-phosphate. As a result, the glycogen chain becomes shorter. This is the process of phosphorylasis of glycogen. And the enzyme which is responsible for this reaction is glycogen phosphorylase. Now here is an overview of the phosphorylasis of glycogen. We had already discussed that, that glycogen contains glycosidic linkages. Whenever phosphate is added, then this phosphate replaces one end of glycogen and it converts the glycogen into glucose 1-phosphate. And glucose 1-phosphate is an energy efficient compound. Okay, this is the process of phosphorylasis of glycogen. Going further, and this process is the first step of glycogenolysis. Now the question arises here is, what is glycogenolysis? It is the process in which glycogen is converted into glucose. We know that glycogen is the storage form of energy in animal cell. Whenever we need energy, the glycogen is converted into glucose, and this process is known as glycogenolysis. And phosphorylasis is the first step of glycogenolysis. And the enzyme which is responsible is glycogen phosphorylase. What does glycogen phosphorylase does in this reaction? It catalyzes or it breaks down the bonds, okay? Whenever phosphate is added to glycogen, it replaces the sugar units from non-reducing end of glycogen branches. As a result, the glycogen chain shortens. It is a more energy efficient process than hydrolysis because it directly produces glucose 1-phosphate. 1-phosphate which can enter glycolysis without needing an additional phosphorylation step. Now question arises here is, why this process is known as energy efficient process? Okay, the answer is that because during this process glucose 1-phosphate is formed which readily convert into glucose 6-phosphate and it generates ATP. As a result, This process is known as energy efficient process. Okay. Now let's describe phosphorylasis of starch. Phosphorylasis of starch is the process where starch is broken down using inorganic phosphate instead of water. The starch phosphorylase enzyme cleaves the alpha-1,4-glycosidic bonds at the non-reducing end of starch or glucan chain. We know that in starch is the main storage form of energy in plant cell. There is alpha-1,4-glycosidic bonds in starch molecule. Whenever phosphate is added to starch, these bonds break down and starch is converted into glucose 1-phosphate. This is the process of phosphorylasis of starch. Now here is the overview of the reaction. The alpha-1,4-glycosidic linkages are broken down due to the addition of phosphate group, and as a result alpha glucose 1-phosphate is formed. Okay, this is the product of phosphorylasis. Continuing further, it is an exolytic process. Now question arises here is, what is exolytic process? Okay, it is enzymatic degradation in which complex molecules are broken down and smaller units are replaced by the reagents. Now it is also an exolytic process, meaning it removes sugar unit from non-reducing end of starch molecule. We know that starch molecule consists of both ends, reducing end and non-reducing ends. Whenever phosphate is added to starch, then phosphate removes sugar unit from non-reducing ends. That's why this process is known as exolytic process. And this reaction is also reversible. Whenever we need energy, the starch is converted into glucose 1-phosphate, and whenever there is no need of energy, then glucose 1 phosphate glucose 1-phosphate again convert into starch. That's why this process is known as reversible. Okay, products are converted into reactant and reactant are again converted into products. This is known as reversible reaction. Now we will discuss the importance of phosphorylasis. Why phosphorylasis is important for living organism. The first point is that it produces glucose 1-phosphate which directly enters metabolism without using extra ATP. Means we had already discussed that this is an energy efficient process because it produces glucose 1-phosphate which readily converts into glucose 6-phosphate and it generate ATP. Okay. The second importance is that it helps release store energy quickly from molecules like glycogen or starch. We know that glycogen and starch are a storage form of energy in animal and plant cells respectively. Okay. So through this process energy is released from glycogen and starch. The third point is, in animals, glycogen phosphorylases provide energy during exercise. We know that during exercise more energy is needed. Okay, so through phosphorylasis, glycogen is converted into glucose to generate ATP. Continuing further, it helps break down starch to release energy during germination or growth. We know that in plant, during germination or growth, more energy is needed. So, phosphorylases provide energy during the period of germination and growth. Unlike hydrolysis, phosphorylasis conserves phosphate group. It conserves more phosphate groups which are needed for glycolysis and Krebs cycle. So these are some importance of phosphorylasis. Okay. Now I also had added some reference book for my audience. The first book is Lee Hanger Principles of Biochemistry by David L. Nelson and Michael M. Cox. Which the edition is 4th edition 2005, page number 540. Okay, the second book is Biochemistry by Leuberts Tyler, chapter page number 610. Now here are some MCQs related to the topic. Feel free to answer the MCQs in comment box. The first MCQ is that phosphorylasis of glycogen produce glucose 1-phosphate. What is the significance of this reaction in muscle cell? Option A, it generates ATP directly. Option B, it bypasses the phosphofructokinase one step, allowing for faster glycolysis. Option C, it produces glucose that can be released into the bloodstream. Option D, it activates glycogen synthase. Now you should answer these MCQs in comment box. The second MCQ is what is the product of starch phosphorylases catalyzed by starch phosphorylase? The option A is glucose, option B is glucose 1-phosphate, option C is glucose 6-phosphate, option D is maltose. Now you should feel free to answer these MCQs in the comment section. Hope so you will understand this topic. And thank you for watching. Thank you for listening.